Happy day everyone. Today in this video we are going to see about a new chapter which is Cell, the Unit of Life from class 11. We have covered the previous chapter in a simple way and I have given tricks for each and every chapter. Please check out the old playlist. And from this video we are going to introduce a new concept, that is, after completing each and every page of an video, I will be giving you some set of questions like that. So once this page is completed, you can try answering this question, and if you are unable to do, you can revise it from the NCERT point of view and then you can do it. Once it is done, you can resume the video and pause on to the next page. So in this way you will be completing the chapter with me, like instead of seeing whole video and you will forget the chapters concept what we are discussing. Instead of that, just see one video and revise it in the NCERT and try answering these questions. These are very simple questions. More than n number of questions can be taken in a single page, but I have given few set of questions only for the practice. So hope this will be helpful for you. Let's get into the video. To start about what is a cell? A cell is the smallest unit which can be show independent existence. So that concludes that anything less than a cell cannot be independent existence. You are getting the point, right? So to talk about the unicellular existence, we have yeast, which is a unicellular organism. These are the example, and it should have the essential functions of life. It can perform the essential functions of life. Obviously in case of yeast, this is a unicellular organism, which means it's made up of only single cell. Still it can perform all the essential functions of life. Now to talk about cell theory. Before talking about the cell theory, the Skildon and Swan firstly find out two things. That is, Skildon said that plants and animals are made up of different kind of cells and these cells form the tissue. And Swan found out that there is a membrane covering the cell and the body of an organism is made up of cell and product of cell. These are acceptable, right? And this membrane is nowadays known as the plasma membrane. And together, after finding this, they proposed a theory called cell theory. In the cell theory, Rudolf Virchow made some modifications, okay? Because whenever some theory happens, we have some changes, we have some improvement. Like the way Rudolf Virchow modified the cell theory. He said that the cell divides and new cells are formed from the pre-existing cells. Because in the old theory, they didn't explain how the new cells are formed. So he gave a conclusion, the cell divides and new cells are formed from the pre-existing cells only. And based on this, the cell theory have two main points. One is cells and products of cell. It is an organism is made up of cell and the product of cells. And the new cell arises from the pre-existing cell. This is the concept of cell theory. Now, we will talk about an overview of a cell. Mainly, the brain of the cell is the nucleus, right? And this nucleus may be of two types. Based on the nucleus, the organism are of two types. One is if the nucleus is membrane bound, that is, if it is covered with nucleus, it is called as eukaryotic. But if it is lacks the membrane, then it is called as a prokaryotic. So this is the difference between eukaryotic and prokaryotic animal. Nucleus contains chromosomes and chromosomes contain genetic material. This is the basic concept, as you all know. Now we have cytoplasm. In the cell, cytoplasm will be there. In the cytoplasm, we have this another organelles. So the cytoplasm is a semi-fluid matrix, okay? Matrix or like area which covers matrix, and is the main arena of cellular activity. This is where the cellular activities happen and the chemical reactions happens within the cell in the cytoplasm, which keeps the cell as living state, okay? This is an important point. To keep the cell in a living state, chemical reactions happen that takes place in the cytoplasm. And the eukaryotic, we talked the eukaryotic or membrane bound organelles. The eukaryotic will be having the membrane bound organelles. In case of nucleus also we talked, right? So what are these membrane bound organelles? Let's see. Endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies and vacuole. So which means in the prokaryotic it lacks all these organelles, right? Eukaryotic means it contains membrane bound organelles and membrane bound nucleus. So these are the membrane bound organelles. So in case of prokaryotic, it will lack all these organelles. And we will have something about called ribosome. It will be present in both eukaryotic and prokaryotic. And these ribosomes are seen in cytoplasm, chloroplast in case of plants, mitochondria and rough endoplasmic reticulum. We will talk about the types of endoplasmic reticulum in the further pages. Now, in the animal cells, we have something called as centriole, which is not present in the plant. About this centriole also we will talk in depth, and these centrioles are mainly helpful in the terms of cell division, and it is a non-membrane bound organelle. And as we all know, the cells are of different types and different size. Let's see what is the smallest cell. The smallest cell known till now is the mycoplasma, and the largest one is the ostrich cell, and these are like measurements of bacteria and RBC. So try to have a look at all these values. So in the first, we have discussed what is a cell and the topics on cell theory and the overview of cell. Now we will discuss the questions. Cell and product of cells, that is, the organism is made up of cell and product of cell. Who given this statement? The next question, what is the smallest cell known? What is the length of RBC? Where does the chemical reaction takes place in the cell? Name the membrane bound organelles of the cell. Centriole is a membrane bound or non-membrane bound organelle, as well as vacuole is a membrane bound or non-membrane bound organelles. If you are able to answer these questions, let's get into the next page. Now we will start the discussion with prokaryotic cells. We all discussed about what is the prokaryotic cells, which is not having the well-defined nucleus or not a membrane-bound nucleus. Okay, so some examples of these prokaryotic cells will be bacteria, blue-green algae, mycoplasma, and pleuropneumonia-like organism. This is the expansion of this pleuropneumonia-like organism. Now, so whenever we say prokaryotic cell, bacteria is the common example or like worldwide example. This differs in this. We have 4 types based on the shapes. What are they? Bacillus, coccus, vibrio, and spirillum. In case of bacillus bacteria, we have rod-shaped cells, and in case of coccus, spherical. In case of vibrio, it will be in comma, and in case of spirillum, it will be of spiral. Okay, these are the 4 types of bacteria depending on their shape. Now, what are the unique characteristics of this prokaryotic cells while comparing with the eukaryotic cell? It has cell wall and cytoplasm and no well-defined nucleus, and it has a naked genetic material as the nucleus is not bound by any membrane, and it has some extra circular DNA which is called as a plasmid. Outside of the genomic DNA there will be a small circular DNA which is called as the plasmid. It gives unique characters such as resistant to antibiotics. These kind of unique characteristics are given by plasmids. Okay, which is a small circular DNA. And apart from that, the prokaryotic cells have ribosomes, inclusions, mesosomes. These mesosomes are specialized differentiant form of cell membrane. Okay, these are present in the prokaryotic cell. Now, we talked about the different types of bacteria right, and we have 2 types of bacteria, which is gram positive and gram negative. This is based on whether they take up the gram strain. If they take up the gram strain, okay, these are some scientific way of testing some organisms. Like for each specific organism, we will some kind of strain, and based on their reaction, we will be dividing it. So one is gram positive and gram negative bacteria. Okay, like have this in mind. Now we are going to discuss about the cell envelope under this modification. This cell envelope contains of 3 layers. Okay, what are they? Gycocalyx, cell wall, and plasma membrane. These 3 acts as a single unit separately, but together, it acts as a single unit together. Okay, so what are the 3 layers? Gycocalyx, cell wall, and plasma membrane. And the gycocalyx is the outer one. This gycocalyx is of 2 types. Either if it is of loose sheath, it is called as the slime layer. If it is of thick and tough sheath, and it is called as a capsule. So what are the types of gycocalyx? It can be of loose and it is called a slime layer, and it can be thick and tough and it is called as the capsule. Right. Now we will see some of the function. To talk about the function of the cell wall, this part it gives the shape of the cell. So the shape of the cell is decided by the cell wall, and it will give the structural support, and it also prevent the cells from bursting and collapsing. Now to talk about the plasma membrane, it has a semi-permeable nature, and it also interacts with the outside world. And we have some specialized structure called mesosome, which we have discussed here. These are nothing but the extension of the plasma membrane, okay, and these extension can be in the form of vesicles, tubules and lamella, okay. There are three types of form for the mesosome, and the functions are they can helpful in the cell formation, cell wall formation, DNA replication, distribution of daughter cells, respiration, secretion process. Also, it will increase the surface area of the plasma membrane. Now, apart from this, we have some specialized structures called chromatophores, which are seen in cyanobacteria kind of organism, and these chromatophores are contain the pigments. Now, if the bacteria is motile, and it will contain some specialized structure called flagella. So the bacteria can be either motile or non-motile. If it is motile, it contains flagella, and this flagella has three parts, which is filament, hook and basal body. Now, among these three, the filament is the longest structure. To continue with that, apart from the flagella, we have some structures called pili and fimbriae, but these don't help in the motility, okay, only flagella will help. This pili are of tubular structure and fimbriae are of small bristle-like structures, and these are made up of special protein. In case of fimbriae, these are nothing but the fibers, and they help the bacteria to attach to the rock or host wherever it wants, okay. Now we will see the questions for that page. So what are the types of bacteria based on the shape? Extension of plasma membrane is called as types of glycocalyx. Okay, which part of the cell prevents it from bursting and collapsing? Which is the longest part in the flagella? This one we recently discussed. And what are the layers of cell envelope? The three layers which we discussed. I hope you are able to answer all these questions. Now let's continue with this page. Now we will talk about ribosome and inclusion body. In the previous video, we have talked about the prokaryotic cell and what are the cell envelope present and what are the modification and their function. Now we will talk about the ribosome and inclusion body. The prokaryotic ribosomes are present in the plasma membrane. So P ribosome, prokaryotic ribosome, and it starts with plasma membrane. So PP, okay, remember that. And these ribosomes are of 15 nanometer to 20 nanometer in size, and they are made up of two subunits, which are 50S and 30S, and together it is the 70S plus prokaryotic ribosome. So whenever they are talked about that, what is the ribosome present in the prokaryotic, you should say 70S. If they ask about the subunit, it is 50 and 30, okay, not 20. Now these ribosomes are the site of protein synthesis. This is a really very important point. The ribosomes are the site of protein synthesis. And we have something called as polyribosomes or polysome. These are made when the several ribosomes, that is many ribosomes, are attached to single mRNA. Know this word. What is a polyribosome or polysome? Several ribosome plus single mRNA together form this. And these ribosomes translate the mRNA into protein. We have the central dogma, right, where we will talk about all this in the 12th chapter. We will discuss this later. So, what is the function of polysome? Is to translate the mRNA into protein. Are we good with the prokaryotic cells? Okay, we have another topic here. It is an inclusion bodies. So, the reserve food materials in cytoplasms are formed in the formed in the form of inclusion bodies, okay. In cytoplasm, the reserve food materials are stored in the form of inclusion body. These are non-membrane bound organelles. So to talk about the examples, phosphate granules, synophisian granules and glycogen granules. And we have gas vacuoles which are present in few organisms such as blue-green bacteria, purple and green photosynthetic bacteria. So with this we are concluding the prokaryotic organism. Now we will move on to the eukaryotic. So what are the eukaryotic cells? Protists, plants, animals and fungi. So apart from Monira, we will classify all the organism into eukaryotic cell. Now the eukaryotic cells will have nucleus plus nuclear envelope, which in prokaryotic it won't be, okay. And apart from that, we have locomotory and cytoskeletal structures in case of eukaryotic cell. Now we will see the major difference between the plant and animal cells. So the plant has cell wall, the animal cell don't have. It has plastid, which lacks in animals. It has large central vacuole, it is not there in animals. But in case of animals, we have centrioles, which is not present in plant. We have discussed already about the centriole. It will help during the cell division. We will discuss about this further later. Now we will see the questions for this page. So the questions will be, what is the size of ribosome present in the prokaryotic cells? Site of protein synthesis, which translate the mRNA into proteins. In cytoplasm, the reserve food materials are stored in the form of dash. So centrioles are present in what? Plants or animals. Several mRNA plus several ribosome is called as polysome. Is it true or false? Now we'll move on to the next page. So in the last page we have discussed about the general things about the eukaryotic. Previously we have discussed about the prokaryotic cell. Now we will talk in detail about the eukaryotic cell. Okay, now firstly we will start about the cell membrane. In earlier days, it was said that the cell membrane is made up of lipids and these are arranged in bilayer, that is 2 layers, in which it has 2 parts. One is polar head and the next is called the hydrophobic tail. So phobic generally means fear. So hydrophobic means it is fear of water. That you can understand, right? And so these together it is called as a lipid. Lipids are arranged in bilayer. It has polar head and hydrophobic tail. So these are arranged in such a way that the polar head connects with the outside water or the cavity, and the hydrophobic tails are saved. Okay, this is the specialized structure of it, and these lipid component of the cell walls are mostly contains phosphoglyceroids. Okay, this is an important point. The lipid components of the cell wall mostly made up of phosphoglyceroids. And after some times it is known that apart from the lipids we have protein and carbohydrates also in the cell membrane, and the amount of protein and lipids are different in each cell. To talk about in RBC, we have 52% of protein and 40% of lipid. Okay, now depending upon the ease of extraction, these membranous proteins are divided into 2 types, which are peripheral and integral. Peripheral means it lies on the surface of the membrane, so it can be easily extracted, right? And the another is called integral, which is partially or totally buried. Now after these discoveries, there is an important discovery done by Singer and Nicholson in 1972. They called it as the fluid mosaic model. This model says that there is an quasi-fluid nature for lipids. Okay, the lipids have a quasi-fluid nature which helps in the protein's movement. This quality, or it is measured as fluidity. Okay, the quasi-fluid nature of lipid is measured as the fluidity and it helps in the protein movement. And one of the most important function of the plasma membrane is the transport of molecules. This transport of molecules can be done in different terms, either passive transport, in which no energy is needed. It will be simply transported to one place to another. Okay, and we have simple diffusion. This is done for neutral solutes. Simple diffusion is when it is moving from higher concentration to lower concentration. It is easy, so no energy required, and it is called as a simple diffusion. So same when from higher concentration to lower concentration, water is moved. Okay, in case whenever the molecules are moved from higher concentration to lower concentration, it is called as simple diffusion. But if water is moved, it is called as osmosis. Okay, movement of H2O by diffusion is called as the osmosis. Now active transportation is where the energy is required, because here it will move from the lower concentration to higher concentration. Generally, if you assume putting a thing from higher to lower is easy, but making it towards the high, it is tougher, so it needs some energy. So this is called as active transport, and the example will be sodium potassium pump. Now we will talk about the cell wall. We talked about the cell membrane. Now we will talking about the cell wall. These cell walls are non-living. Cell walls are non-living and rigid, solid kind of thing, and it covers the plasma membrane in plants and fungi, because cell walls are absent in animals, as you know now. Now we will see the cell walls are made up of different types of components in terms of different kinds of plants. In terms of algae, it contains cellulose, galactans, mannans and calcium carbonate, which is a mineral. But in case of other plants such as pteridophytes, bryophytes, these are the other plants, right? It is made up of cellulose, amylose, pectins and proteins. These are the different types of component present in them. And we have middle lamella, which separates the which glues or connect the different neighboring cells. With help of different neighboring cells, with the help of middle lamella, the different neighboring cells are connected together. Okay, this is the use of the middle lamella, and it is made up of mainly calcium pectate. And between the cell wall and middle lamella, okay, what is the middle lamella? Which connects the different neighbor cells. So between the cell wall and middle lamella, we have plasmodesmata, which connects the cytoplasm of neighboring cells. You are clear with these terms? Middle lamella neighbors the cells together, and this between cell wall and middle lamella, we have plasmodesmata, which connects the cytoplasm of neighboring cells. Okay, now we will see the question for this page. The first question will be, in the lipid bilayer, which interacts with the outside world, outside cytoplasm. What is the percentage of protein in RBC? Fluid mosaic model, I have given in short form like FMM. Fluid mosaic model was found by whom and in which year? Give an example for active transport. Pectins are found in algae cell wall. Is it true or false? Which separates the cell wall and the middle lamella in eukaryotic cells? I hope you revised this page. Let's move on to the next. So we are talking about the eukaryotic cells, in which we have discussed about the cell membrane, cell wall. Now we will talk about the endomembrane system. What is this endomembrane system? The endomembrane system, in the organelles in these are like coordinated in their function. That's why it is specifically called as an endomembrane system. And endoplasmic reticulum, Golgi complex, lysosome and vacuoles. These are the 4 organelles inside this system. Now we will talk about each in detail. So talk about the endoplasmic reticulum. These are the network of tiny tubular structure found in the cytoplasm. Okay, and these divides the intracellular space. Intra means inside, so cellular means intracellular space. Divide the intracellular space into 2, which is luminal and extraluminal. Luminal means inside ER. Whatever the space inside ER is called as the luminal, and what are the space covered in the cytoplasm are called as the extraluminal. And these endoplasmic reticulum are of 2 types, which is rough endoplasmic reticulum and smooth endoplasmic reticulum. In terms of rough endoplasmic reticulum, it is characterized by the presence of ribosomes, small dot structure if you see in the photo. So, and it is the place of protein synthesis and secretion, because we already, in talking about the ribosome, we said that the ribosomes are the site of protein synthesis. So here we have ribosomes, so it means rough endoplasmic reticulum synthesis the protein and also secretion. Now it has connected with the outer membrane of nucleus. What are the characteristics of rough ER? Presence of ribosome, protein synthesis and secretion connected with outer membrane of nucleus. We will discuss about this later while we discuss about the nucleus. Now to talk about the smooth endoplasmic reticulum, there is no ribosome and it helps in lipid synthesis, and in case of animals it synthesizes some steroidal hormone which is a lipid like. Okay, these are the 2 types of endoplasmic reticulum. Now we will talk about the Golgi apparatus. This is named after the Camillo Golgi because he is the one who found this or like observed these material. These are like generally densely strained reticular structure. Okay, these are like densely strained, but in case of endoplasm, these are like tiny tubular structure. So these are the difference about each organelle in this shape. And these Golgi bodies contain cisternae. These are nothing but some flat or disc shaped sacs. Okay, flat or disc shaped sacs, or it is called as cisternae, and these are of 0.5 microns or micrometers to 1 to micron. So 0.5 to 1. This is the size of the cisternae. These are arranged especially near the nucleus with either convex or cis phase, concave or trans phase. Okay, it has two phases. One is convex, and near the nucleus it may be attached either with this convex or cis phase or concave or trans phase. The convex is the forming phase and concave is the maturing phase. These both are entirely different, but they are interconnected. Okay, this convex and concave phase are entirely different, but they are interconnected. These Golgi bodies perform the packaging of material. The main function of the Golgi body is to package the materials. This packed materials will either sent to the intracellular within the cell or to the outside cytoplasm or any other organelles. The material to be packed in the form of vesicles from the endoplasmic reticulum will be fusing with the cis phase. You are getting the point, okay? So the materials which are need to be formed in the form of packed in the form of vesicle will be joined at the endoplasmic reticulum, and it fuse with the cis phase of the Golgi apparatus, and it move to the trans phase. And this becomes the reason why the Golgi body and the ER, that is endoplasmic reticulum, are close in structure. Now let's see how this is happening. Firstly, this proteins comes from the endoplasmic reticulum, will be modified from the cisternae. Then it will be do the packaging things. Then it will be released from the trans phase. This is how the packaging of materials takes place in the Golgi body. Now we will talk about lysosome. So these lysosomes are formed when the packaging process is happening in Golgi apparatus. We talked, right, the Golgi apparatus is mainly for packaging of material. So whenever this is happening, during that process lysosomes are formed. These lysosome vesicles are high in hydrolytic enzyme. Okay, they contain more hydrolytic enzyme and hydrolysis such as lipases, proteases and carbohydrates. Hydrases. As you all know, these are useful for the digestion of the specific enzymes. In case of lipases, it will digest the lipase. Okay, so then it will activate the acidic pH. It has the ability to activate the acidic pH. These are all about lysosome. Now we will talk about the vacuoles. The vacuoles are nothing but the membrane bond space. Okay, these are some space which are found in the cytoplasm. So what are the materials found in the vacuoles? It's a simple space, right? So it contains water, sap, excretory products and also not useful product for the cell. And these are single membrane. Okay, these are made up of single membrane. Whether the organelle is made up of single membrane or double membrane, how to memorize it, I have given a trick video for that. You can go and watch it. So it will be easier for you to, instead of memorizing each and every one. Now this single membrane, it is called as tonoplast. In case of plants, 90% of the volume is occupied by this vacuole only. So it will be large in size. Now the concentration in the vacuole is higher than in the cytoplasm. It's because in the vacuole it will be the transport facilities, it will be in the against the gradient. What is against gradient? It means like it moves the low to higher, that is against the gradient. That's why the concentration in vacuole will be more in the it is more than the cytoplasm. And to talk about the different type of vacuoles, in case of amoeba we have contractile vacuole which help in excretion. In case of protist we have food vacuole which helps in engulfing the food. It will just engulf the nearby food. Okay, here we have discussed about endomembrane system and Golgi apparatus, lysosome and vacuoles. We will see the questions for this. The first question, what are the organelles found in endomembrane system? Which is the site of lipid synthesis? Give the size of cisternae found in Golgi apparatus. Convex phase is called as forming or mature phase. What is the membrane of vacuole called as? What is the percentage of vacuole in plants? Food vacuoles are found in. I hope you have able to answer all these questions. If not, revise it once along with the NCERT, then we will move to the next page. So lastly, we have discussed about the endomembrane system and the organelles inside it in detail. Now we will talk about the mitochondria. This mitochondria will be different in number and size in each of the cell depending upon the need, and these are double membrane. So I have made a trick video, as I said before, so to memorize what are the organelles which are of double membrane and single membrane and membraneless organelles. Okay, so please do watch that video. Now we will talk about the mitochondria in further. So this in double membrane, one of which is inner and other is outer. This inner membrane will divide the lumen of the mitochondria into two compartments furtherly. One is outer compartment and inner compartment. Okay, so there are two membranes, which is inner and outer. This inner membrane will divide the lumen into outer component and outer compartment and inner compartment. This inner compartment is called as the matrix. Now this outer compartment will form the limiting boundary of the organelle, but the inner compartment forms the number of infolding, which is called as cristae. In case of Golgi apparatus, we talked about cristae, but here it is cristae. Okay, so outer compartment forms the limiting boundary, like stopping, but the inner compartment forms the inner folding. It is called as the cristae, and it helps in increasing the surface area. And these are towards the matrix. Matrix are nothing but the inner compartment, right? Now these mitochondria are the powerhouse of the cell because they has the aerobic respiration site. They produce ATP, that is energy. That's why it is called as the powerhouse of the cell. Apart from this, we have some things in the matrix which are nothing but the single circular DNA, few RNA, and 70S ribosomes, and also synthesis of proteins is also taken in the mitochondria. These are the functions and characteristics of mitochondria. Now to talk about plastids. Plastids are found in plant cells and in euglenoids. Based on the types of the pigments, there are three types. One is chloroplast, next is chromo, and another is leucoplast. These chloroplasts are containing the chlorophyll, that's why it's named as chloroplast, and carotenoid pigments, and these are help in photosynthesis. Obviously, whenever you say chlorophyll, you should remind immediately about photosynthesis. Now to talk about the chromoplast, it has the fat soluble pigment, carotenoid pigments such as carotene, xanthophylls, and it gives the color to the plant such as yellow, orange and red. Now to talk about the leucoplast, it is colorless. In case of leucoplast, it is colorless and it stores nutrients in three types. One is amyloplast, eleoplast and alluroplast. This is in the form of storing the food. The amyloplast will store carbohydrates, which is starch, and eleopols, like as the name indicates, it stores oil and also fat, and alluroplast stores protein. These are about the types of plastid. And to talk about the chloroplast, these chloroplasts, as discussed, it contain chlorophyll, and these are generally found more in the mesophile region. These mesophiles are nothing but lens or oval or spherical or ribbon shaped structure found in the plants, and these mesophiles are differ in numbers. In case of chlamydomonas, it may be 1, but in case of green algae, it may be 20 to 40. Now these are contain double membrane, and I have trick video for that, in which the inner membrane, the space limited by the inner membrane, is called as stroma. This stroma contains flash sacs. These are called as thylakoids. Okay, the place limited by the inner membrane in the chloroplast is called as stroma. This stroma contains flask or flat sacs which are called as the thylakoids. These thylakoids will be stacked or like arranged in the like a piles of coins, and that is known as the grana or intergranal thylakoids. So thylakoids together form as a stack which is known as the grana. Okay, so between thylakoids of different grana, Okay, what are the thylakoids? Thylakoids is single structure, single flat structure. Grana is when thylakoids are arranged together. So between thylakoids of different grana, there will be stroma lamellae. Say this is one grana that is arranged in like a pile of coins. This is for example I have drawn like that, and this is another grana. So between the thylakoids is these single. So between the thylakoids of the different grana, there will be a stroma lamellae. This is what it detects. And also in thylakoids we have chlorophyll pigments. Okay, so the chlorophyll pigments are present in the thylakoids of the chloroplast. To continue with the chloroplast, we have another structure called lumen within that, which is nothing but the space which is enclosed by the membrane of thylakoid. The space which is enclosed by the membrane of thylakoid is called as lumen. The stroma. What is stroma? Try to revise. It is a space which is limited by the inner membrane of the chloroplast. Right. Inside the stroma, we have synthesis of carbohydrates and protein, that is enzyme required for the synthesis of carbohydrates and protein, small double-stranded DNA, and also ribosome. These ribosomes is of two types here. One is that is 70S, which is found in chloroplast, and another is 80S, which is found in the ribosomes. Are we clear with this passage? Now we will see the questions, but we will complete these also together and we will see the questions for the previous page and also for this page. Now to talk about the ribosomes, these are firstly discovered by George Palade in the year 1953. Okay, right. They are composed of RNA, which is ribonucleic acid, and proteins, and are not surrounded by any membrane, membraneless organelles. Okay, and in case of eukaryotic, we have 80S ribosomes, but in case of prokaryotic, we talked about 70S. What is this? S denotes Sudbeck's units. Okay. Now we will talk about cytoskeleton. These cytoskeletons are nothing but the filamentous proteinaceous structure, networks of filamentous proteinaceous structure found in the cytoplasm. So these together called as the cytoskeleton. They have the mechanical support, motility, and also maintain shape. Now we have cilia and flagella. Cilia and flagella are the locomotory organs, we can say, and these can be small. I mean, cilia are the small one, flagella are the large one. These are nothing but the hair-like a minute hair-like soft projection outgrowth. These are covered with the membrane, and we have axonum, which is nothing but the core part. The core part of the cilium or flagellum is called as the axonum, and it has 9+2 array. So what is this 9+2 array? We have 9 doublets which are arranged radially. Okay, radially peripheral arrays, but in central we have 2. So 9+2, 9 outside and 2 in central. That is called as the 9+2 array. These in central we have 2, right? These are connected by bridges, and the central tubules are covered by central sheath. These are connected by bridges and covered by central sheath, and these central sheath will be attached to the one of the peripheral tubules. It will be in doublets, right? It will be in doublets. It attached to one of the peripheral tubules like this. 2 are there. It will attach to one in the terms of radial spoke. Okay. The central sheath, which is surrounded by the central tubules, are attached to the peripheral tubules in terms of radial spoke. And these peripheral tubules too are connected by the linkers. Okay. I'm sorry for confusing you. These peripheral tubules are connected by linkers, but the central tubules are connected by the bridges. Central and peripherals are connected by radial spoke. I hope you are clear now. And where are these cilia and flagella coming from? These are emerging from something called as basal bodies, and these basal bodies have centriole-like structure. Now we will talk about the centriole. Centrosome and centriole. The centrosome is an organelle which contains the centrioles. These centrioles are cylindrical in structure, and in centrosome we have 2 cylindrical structures, that is centriole, and they are arranged perpendicularly. As like in cilia and flagella, here also we have 9 peripheral fibrates, but these are in triplets. In case of that, we talked about doublet. Here it is triplet, and these are nothing but the tubulin proteins. And the central part of this is called as the hub. Okay, this hub is also made up of proteinaceous layer, and this hub and tubulin proteins, that is peripheral fibers, are attached by radial spoke. This is about the centriole and the centrosome. Now we will see the questions for the previous and this page. The first question will be, what is cristae? The next, which is called as a powerhouse of the cell. Ribosome in mitochondria are of 70S or 80S. Alveolus stores what kind of materials? Chromoplast contains which pigments? What is grana? Ribosome in eukaryotes 80S or 70S. How are the peripheral tubes or interconnected? Central proteinaceous part in centriole is called as. I am not discussing the answer because just now we are revising the concept, so I thought you will be able to find the answer also. Now we will move on to the next page. So we have discussed about most of all the organelles in the eukaryotic, like endoplasmic reticulum, mitochondria, plastids, what else, ribosome, vacuoles, lysosome. Now we are talking about the last and the main important one, which is nothing but the nucleus. Now let's get into the video. Nucleus is first discovered by Robert Brown in 1831. Robert Hooke found the dead cell. Okay, he is the one who find the dead cell first. That is Robert Hooke, but nucleus is found by Robert Brown. Now we will discuss the nucleus with the structure. Basically, this nucleus is made up of inner and outer layer, and it is the brain of the cell. Okay, it has the activity, and it has the DNA. So it is called as the brain of the cell, and it is made up of two layers. One is inner layer and another is outer layer. Within this inner and outer layer, there will be a space, right? It is called as the perinuclear space, and the size is of 10 to 15 nanometer. So we have two layers of the nucleus, which is inner and outer. The space is called the perinuclear space. Then we have nucleopores, which are there between these layers. Whenever the fusion of these layers is happening, say here the fusion has happened, so we have some space. This space is called as the nuclear pores. Okay, these pores are called as the nuclear pores, and these are helping transporting the RNA and protein in either side, that is from nucleus to the outside, from outside to the nucleus. Okay, that is the function of this nuclear pore. And we have nucleoplasm. So this is that surrounded area in the nucleus is called as a nucleoplasm or nucleomatrix. Within this nucleomatrix, we have nucleolus, which is the site of ribosomal RNA synthesis. Okay. Now, ribosomal RNA synthesis is done in the nucleolus, and in each cell the nucleolus is differ. Okay, if it needs the high protein synthesis, the nucleolus will be in high in number. Okay. Now we have something called as chromatin in the interphase nucleus. What is the interphase nucleus? The nucleus which are not dividing, it is called as interphase nucleus, and in that we have the chromatin structures. These chromatins are first found by Fleming. Okay, Robert Hooke found the nucleus, but chromatin was found by Fleming. He found it when the nucleus is stained by the basic dye. As we discussed about the gram positive and gram negative bacteria, here also some basic dyes are used to stain it, and from that we found out this chromatin. These chromatins are nothing but the network of nucleoprotein fibers, and it has the DNA histones. Histones are the basic proteins, we could say. This and all we will be talking in detail in the 12th chapters. Histones and non-histones and RNA. These are the materials found in chromatin. So this is a general view which I have simplified. What are the things given in NCERT? You have to Remember these things, that will be enough. That is inner and outer membrane, perinuclear space, nucleopores, nucleoplasm, nucleolus, chromatin. And the main part is this endoplasmic reticulum will be attached to the nucleus, and also it bears the ribosome. So as you all know, when it bears ribosome, it is the rough endoplasmic reticulum, because we have the ribosomal RNA synthesis happening here. That's why this endoplasmic reticulum is closely attached with the outer layer of the nucleus. Now we will talk about some few information given about DNA. So the DNA in the single human cell will be of 2 meter long, and it will be like spread out with the 23 pairs of chromosomes. As you all know, we have 23 pairs of chromosomes, so 46 in total. So the DNA will be distributed along all this, and it is of 2 meter. And we have some different types of chromosome based on the position of the centromere. We have metacentric, submetacentric, acrocentric, and telocentric. All these things we will be discussing later in the upcoming molecular chapters, okay, molecular inheritance, those kind of chapters. So I have given the basic idea, and we have given about microbodies. These microbodies are nothing but some minute vesicles, okay, and it is found in both plants and animals, and it is a membrane bound organelle. So with this we are completing this chapter. So we will see the questions for this page. Nucleus is found out, discovered by whom? Chromatin discovered by whom? What are the function of nucleopores? Size of perinuclear space. Place of RNA synthesis in nucleus. What is the size of a DNA in single human cell? Does microbodies have membrane or not? So with this we are completing this chapter successfully. I hope all this video will be helpful for you, because I have included the questions in between. I haven't given it in the forms of MCQ because I hope you will be able to answer these questions even without options. So that's why I will make an MCQ video or like Q&A video for this chapter, covering all the topics in a single video also. Keep revising. We'll see you in the next video. Please try to watch these strict videos. That will be really helpful for you. Thank you.